All right, folks, this is a big one. This is probably the trickiest individual subtask of this entire unit. The trick is in knowing the names of a few different proteins and the job they do in replication. So this is going to be Subtask 2. It's found on page 7 of the learning targets. And it basically is 3 essays. The first one, how does replication start? The next one is how does replication work on that leading strand? And then the last one, the longer one, is how it happens on the lagging strand. So to catch us back up, there are going to be a few enzymes and proteins that come up here, and I'm just going to give you their names now. They'll all have a role to play eventually. Topoisomerase, helicase, single-strand binding proteins, DNA primase, RNA primers, DNA polymerase 3, DNA polymerase 1, these chunks called Okazaki fragments, and DNA ligase. Now, the ones up here that are enzymes that are actually going to perform a job for us all end in ACE. So topoisomerase, helicase, DNA primase, DNA polymerase 3 and 1, and DNA ligase are all enzymes that have jobs. The other things up here are other players, other molecules, but they're going to be important. What has to happen at the beginning of replication, this is like the very first thing, very first step. Topoisomerase relieves coiling strain. Imagine a rubber band that you've twisted up and then you snip one of the ends so the rubber band can uncoil. That's what topoisomerase does. Helicase, the little triangle, is going to go down like a zipper and unzip the DNA, separating the two parent strands. Single-strand binding proteins make sure that the strands don't reconnect, or what we would call re-annealing. This is going to create 2 strands. One that can be built, the daughter strand, can be built towards helicase, and one that has to be built away from helicase. Towards helicase is the leading strand, and this one's pretty short and sweet. If you are the leading strand, what's going to happen very first is there's going to be a little chunk of RNA that you need to begin at. That's going to be laid down by DNA primase. It's a little chunk of RNA connected to the DNA. It's shown in red here. And that's going to be a starting point. What that's going to mean is there's this little enzyme shown here with this little yellow circle. That enzyme is going to float up and connect right on the RNA. From there, it's going to go from 5 to 3. And as it goes, it's going to build this green strand right here of new DNA. As helicase keeps going, the strand can keep being built. It adds nucleotides in a complementary base pairing all along this strand, building the new daughter strand from the parent strand template. And that can continue until replication is done, so that's all we have to talk about with the leading strand. An RNA starting point and one enzyme, DNA polymerase 3, that does all of the building. The lagging strand's a little trickier. And it's trickier because you have to build it backwards. So here's how that's going to work. The first thing that's going to happen is this little enzyme, shown here in red, is going to move along the lagging strand and put down little chunks of RNA every so often. Those little chunks of RNA are called RNA primers. They act as starting points. And here, this little enzyme that's doing that laying down is called DNA primase. So now that we've got our little primer set up, this is going to follow helicase, leaving little RNA primers. They're little chunks of RNA that are about 5 to 10 base pairs long. And these are going to be markers for DNA polymerase 3 to start building from. Now from here, things are going to look a little bit like the leading strand. Starting at a primer and going from 5 to 3 backwards, DNA polymerase 3 is going to build DNA. When it gets to the next RNA primer, it stops, detaches, and then moves to the next chunk that needs to be built. So if we go through that whole process with this little leapfrogging, we're ultimately going to get DNA that has been built in these short chunks in between these little starting points. Again, the whole reason we have to do this is we can't just build this way and follow helicase. As more DNA gets unzipped, you have to build that next little chunk backwards. Now, the problem here is that we don't have just DNA up here. We have DNA and RNA, and we're going to have to get rid of that RNA, and that's where a couple more steps come in. Now, these little chunks of DNA that are only on the lagging strand are called Okazaki fragments, discovered by the Japanese scientist, and when you discover something in science, you get to name it. So, all of these little DNA fragments separated by RNA primers are Okazaki fragments. We need to connect those Okazaki fragments and get rid of the parts that are not DNA. So DNA polymerase 1 comes in next. Now, why would 3 be first and DNA polymerase 1 come next? I believe it's because that is the order in which they were discovered. You discovered DNA polymerase 1 first, and even though DNA polymerase 3 happens first in this process, it wasn't discovered until later, so it has the name 3. Now, this one has a name that indicates what it does. It creates DNA, just like polymerase 3, but this one has a very special job. It moves up to the RNA primers, it removes them, and then it replaces them with a little chunk of DNA. So now, when this is done, there shouldn't be any RNA left. It goes up, removes it, and now that RNA has been replaced with a green strand of DNA. Now, in this case, we still have these Okazaki fragments. There's no DNA, or there's only DNA here, but it's not connected. There's that little chunk that still needs to be put together. And that's where our final enzyme is going to come in, and that enzyme is called DNA ligase. This one actually connects nucleotides with a phosphodiester bond. So there's 2 nucleotides here that you can imagine. They just have not been connected 5 to 3 yet. DNA ligase comes in, is going to attach our Okazaki fragments together, and now we have a continuous strand of DNA. And that's the lagging strand. Again, a few more parts when you have to build the DNA backwards. In total, here's what these all do. They all have a role to play. Topoisomerase cuts the sugar phosphate backbone and repairs it to relieve coiling strain. DNA helicase unwinds and breaks hydrogen bonds between base pairs, unzipping the DNA. Single-stranded binding proteins prevent them from re-annealing, or the 2 strands from reconnecting again. DNA primase is going to leave primers, RNA primers, that are going to act as an initiation site for DNA polymerase 3. DNA polymerase 3 does most of our building. It attaches nucleotides and it attaches them in a 5 to 3 direction. DNA polymerase 1 is going to remove the RNA primers and replace them with DNA, again in a 5 to 3 direction. Now, those little chunks that used to be separated on the lagging strand are called Okazaki fragments, discontinuously built sections of new DNA only on the lagging strand. And we connect them by getting rid of the RNA primers, replacing them with DNA, and then finally, DNA ligase attaches the Okazaki fragments together. Once that happens, you should have 2 strands of DNA that have been built from the original. They are fully connected, they should be identical to one another, and replication is done.